Wonderful. Thank you, everyone. Welcome, everybody. Thank you all for coming. My name is Ben Dibbling. I'm Associate Vice Provost for Research and Managing Director of the Penn Center for Innovation. It really is wonderful to be here today. I'm delighted to have the opportunity to welcome so many alumni, colleagues, collaborators, and friends of Penn to what I think is our 10th annual Innovation at Penn event at Wharton, San Francisco. I'm joined here by some of my colleagues, Laurie Ackman, PCI's Chief Marketing Communications and Programs Officer, Sam Civello, who's our Marketing Communications Manager, Carter Caldwell, who directs the Penn Medicine Co-Investment Program, Melissa Lopez, who's an Associate Director in our Ventures team, Jim Bowen, who's the Executive Director of our Corporate Alliances Group, and John Swartley, Penn's Chief Innovation Officer. And for those that don't know, at the Penn Center for Innovation, or PCI, as we call ourselves, our team's focused on identifying and implementing relationships with the commercial sector to facilitate the development of new products and services and to establish new ventures. All of this based on inventions and discoveries that are generated by Penn's outstanding faculty and research scientists. PCI had another productive year, and in fiscal year 2025, we brought in over $500 million in revenue, which ranks us number one nationally for the 5th consecutive year in a row. We executed around 650 commercialization agreements, and again, this reflects the breadth of relationships we're supporting between Penn innovators and industry. We received about 400 new inventions. Again, that's an indicator that Penn faculty and researchers continue to vigorously innovate and pursue research ideas that push boundaries and address global challenges. And Penn spinouts raised over $591 million in funding, despite what I think we can all agree is a challenging funding environment. And I'd encourage you to go to our website, download our year in review, and you can see more of the details and some of the key stories that we've wanted to highlight. Fiscal year 2026 is off to a strong start. Highlights include 2 high-profile acquisitions of Penn startup companies, both in the in vivo CAR-T space. So Capstan Therapeutics was acquired by AbbVie for $2.1 billion. We have Laura Shorver, who's moderating the panel, who was the CEO of Capstan, and also Dr. John Epstein, who is one of the scientific founders of Capstan. And the other company was Infectious Biotherapeutics, which was acquired by Gilead's subsidiary Kite for about $350 million. That said, I want to just spend a minute or two just highlighting 2 really exciting initiatives, the launch of 2 new investment vehicles at Penn that we expect will accelerate the spinning out and success of new ventures based on Penn technology. You might have seen the news on Friday where the launch of the Penn BioNTech Innovative Therapeutics Seed Fund, or PXP Fund, which is a little easier, which is a fund of $50 million, which will make seed and Series A investments in Penn companies developing therapeutics, diagnostics, and research tools. The fund will be managed by Osage University Partners, or OUP, which is a Philly-based venture capital firm. And John Swartley and Jim Bowen were both instrumental on the Penn side in establishing this PXP fund. That news actually follows hot on the heels of an announcement at the end of last year of a complementary fund called the Startup Fund, which will be managed by John Swartley out of his Office of the Chief Innovation Officer, and is backed by a $10 million commitment from Penn, and will be providing initial capital to early stage companies formed around technologies originating from Penn. We've got a really great panel for you again this year, and they'll be discussing precision medicine and how this relates to the development and delivery of targeted therapeutics. It's not a new topic, but we feel it's particularly important today based on the wealth of patient data being generated, the promise of AI, and the news from last year of the high-profile and successful personalized CRISPR therapy that was used to treat a rare urea cycle disorder in an infant known as baby KJ. And this world first resulted from a collaboration between Penn and CHOP, led by Dr. Kira Musunuru and Dr. Rebecca Ahrens-Nicholas. I want to thank the panelists for their participation, Laurie Ackman and her team for putting this event together, Wharton San Francisco, again, allowing us to use this beautiful space, and to our sponsor, the law firm Sal Ewing. So before we get to the panel, it's really an absolute pleasure to introduce our keynote speaker, Dr. Jonathan Epstein, who serves as Dean of the Raymond and Ruth Perlman School of Medicine and Executive Vice President of the University of Pennsylvania for the Health System. And we collectively refer to these entities as Penn Medicine. I could spend a lot of time talking about John's history, but suffice to say, during his time at Penn, he's run his own highly productive research program in addition to holding various leadership positions. He's been instrumental in the growth of Penn Medicine's research enterprise and helping it to become a national and global leader in discovery science and clinical research. We work with Dean Epstein a lot, and we're deeply appreciative of his unwavering commitment to innovation and supporting the development of new medicines to the benefit of the public. And with that, I'll hand over to John.
I'll repeat my thank you to Ben. It's a pleasure to be here, and I want to welcome everybody. Thank you for joining us today. I'll make just a few brief comments before we move on to the panel. Many of you and your colleagues who I've run into over the last day or so here at the JP Morgan conference have asked me with a sort of serious look on their face, how's it going? What's it like to be a dean of an academic medical center, to be a dean of Penn in these times? So I thought I'd make just a few brief comments about what it's like these days. And I can summarize it quickly by saying it's the best of times, it's the worst of times. We're under a lot of pressure. There have been cuts in federal funding, maybe even more significantly, threats of cuts to federal funding that have affected the climate quite significantly, and I think are relevant to everybody at this conference because of the threat to the pipeline in biomedicine more broadly, both the pipeline of talent and the pipeline of ideas. But it's also been the best of times because the kind of advances that Ben was describing, that PCI has so ably helped us to commercialize, they keep coming. And what I've seen, I've been at Penn now for 30 years, I've seen a complete transformation of the culture and of the approach that our faculty and our trainees take towards their work. When I trained, our job was to write papers and get grants and do other experiments that we hope created new knowledge. But I thought, and I think my colleagues thought, that companies would take those ideas forward to make new medicines from them. And that has been completely altered. Now, because of the success that we've seen, our trainees and our faculty think their job is to create new cures. And they have the confidence that they think they can do it. There's no better example than baby KJ, in that story of in 6 months or 7 months, going from the birth of the child to sequencing the genome, to all the necessary studies through non-human primates, to working with the FDA to get approval, to getting a product manufactured, to delivering it to the child. It takes a lot of confidence and a lot of energy and know-how to make that happen. And I'm so proud of the faculty and the trainees who are coming up through the ranks at Penn these days because they know that they can do it. And they're inspired by the successes that they see. So just, you're probably all familiar with this, but I have to brag because I'm here, I have the opportunity to brag a little bit about Penn. You think about the advances that have come out of work primarily at Penn, the advent of CAR T cells, of mRNA as a therapeutic, as gene therapy with Zolgensma and Luxturna, Jim Wilson here today, who's led much of that work, the new Alzheimer's drugs, largely based on work in Virginia Lee and John Trojanowski's laboratory, and the use of CRISPR to correct genetic defects in people. It's a pretty good track record. Something like 45 FDA approvals for new drugs since 2015 or so. Any company would be very proud to have that track record. So that's the best of times part of what I see. And I'll just add to that list, just in the last year, a publication by Lou Chodosh and Angie DiMichele about so-called sleeper cells in breast cancer. Many people with breast cancer get treated, but it comes back in a treatment-resistant fashion afterwards in ways that have been unpredictable up until now. And they've identified essentially a stem cell that resides in the bone marrow that is the cell of origin of the recurrent metastatic disease. And they also came up with ways to keep those cells quiescent. But they weren't happy to do the mouse studies. They did a phase 2 clinical trial in patients and showed that they could reduce the incidence of recurrent treatment-resistant breast cancer. Again, another sign of the culture at Penn that it's not good enough to just write up the research study. They want to take it all the way to clinical trials. And just today, Jim Shorter had a paper in Molecular Cell that shows that RNA nucleotides, range of like 12 to 24 nucleotides in length, could change the phase transitions that take place in TDP-43 and FUS-mediated neurodegenerative disease, and change the aggregates that form in those cells, suggesting that this might be an RNA-based approach for treating these forms of neurodegenerative disease. That work is published, and there's follow-up work that's even more impressive that's being reviewed right now. So, the pace of advance continues, and I urge you to keep your eye on the latest work coming out of Penn. This changing culture changed what I do in my own laboratory. I have for years studied the genetics of congenital heart disease and did very basic work in animal models and was proud to publish those reports. But I looked left and right of me and saw these successes and asked if we couldn't do some things that had more impact and were more translational. And led by students in my lab, Haig Agajanian and Joel Rurik, we did the original work showing that you could generate in vivo CAR T cells with mRNA, which led to Capstan Therapeutics. And I'm delighted that Laura Shawver is here to lead our panel today, and I'll introduce her further in just a few minutes. So I feel this changing culture viscerally at Penn because it's affected my own career and my own laboratory. At the same time, I do think we're at a time of transformation in the biomedical pipeline. I think the world is changing, and it's not going to go back to the way it was, even if there are changes in the administration, et cetera. And I think that even a place as accomplished as Penn at creating new therapies will need to adapt in the future to a new normal and a new world. And frankly, I think this crisis may be an opportunity to think about how we can do it even better. So even though we've done it very well, it's still too slow, and it's still too expensive. We still generate new ideas in academia. One by one, we license them, or we spin out companies. We spend a lot of time with lawyers and negotiating contracts at that step. Then we spend time raising money. Then eventually all those studies are repeated in the private sector because nobody believes what's done in academia, or we didn't do them in an IND-enabling way. And then it's time to do the clinical trials, and we either go to China or to Australia, or maybe we come back to academia and have more negotiations and agreements that lawyers get involved with. Surely we can come up with a system that shortcuts some of those steps in the process. And I'm very eager to talk to people at this conference and in other settings about new models by which we might fund early stage development of new ideas in a way that bypasses and short circuits some of those steps. And there isn't time here for me to go on and on about it, but I'd be happy to talk to people individually about some ideas that we are pursuing that I think will lead to different models of how we generate translatable, translational ideas. So with that, I'll introduce Laura Shawver, who I've had the real pleasure to work with during the period of Capstan Therapeutics. Laura is a serial successful biotech executive with over 30 years experience in the biopharma industry, most recently at Capstan Therapeutics, but prior to that, she was the chief executive officer and director of Silverback Therapeutics, which was also acquired. Before that, president and chief executive officer of Synthorex, which was acquired by Sanofi back in 2020, and she had a series of other positions in biotech and pharma prior to that. She's also, frankly, just a great person, and she cares deeply about the science and about the patients who we ultimately serve, and she's personally invested in philanthropic efforts, creating a foundation called the Clarity Foundation for ovarian cancer research, which recently merged with the Ovarian Cancer Research Alliance. She received her PhD in pharmacology and a bachelor's degree in microbiology from the University of Iowa, and it's my great pleasure to welcome Laura Shawver here to the podium.
Thank you, John. I'll invite my fellow panelists to the stage, and while they're coming up, I'll just say, John, that I'm glad that you brought up the foundational work of Capstan with the science paper that demonstrated that you could actually in vivo engineer T cells. And I'm just going to say that not only did you launch a company, you launched a field. And I think that later this year, the field will show that this really works in people with many different types of B-cell mediated diseases. And you talked earlier about how proud people were of the work that they did. And I really hope when you see those data that you and Joel and Hai are immensely proud, because it really launched a very important field. And of course, I think people know that Capstan stood on the shoulders of many giants, not just Dr. Epstein, but Carl June and Bruce Levine and Drew Weissman. And it was really many years of effort that led to Capstan and the whole approach of in vivo. We just had the privilege and the pleasure to take it a little bit further. And of course, now it's in the hands of AbbVie, which of course can bring it to massive numbers of people that really need this treatment. But thank you for the kind introduction. Okay, so we have a great panel today, and we're going to start here with Jim Wilson. Thanks for being here. And I think people may know that currently you're the CEO of Gemma Biotherapeutics and exec chair of Franklin Biolabs. You've also been doing this a long, long time, like me, and you've been an innovator and a pioneer in the field of gene therapy, all at the University of Pennsylvania. And while there, you've identified, isolated, developed virus-based vectors for in vivo gene therapy. You were responsible for discovering and bringing to bear a whole field of adeno-associated viruses that have become important vectors in novel therapies. And John mentioned some of these, but Glybera, Luxturna, Zolexma. And so the first question to you is, with all that you've brought to the field at Penn, why did you decide to go big with your new startup companies? And why now?
Well, thank you, and thank you, PCI, for asking me to participate in this. We've been partners for the entire time, Jim Bowen and others, John, Ben. Well, I can promise you one thing. When I was a postdoc at MIT, I was not aspiring to be a CEO of a company. so something had to change, and if you'll allow me, I'll give you some perspective on that. I am, in the spirit of full disclosure, I'm unabashedly a supporter of rare diseases. That's what I do. The rarer, the more we lean in. And from that perspective, despite all of the successes we've had at Penn, patents, publications, startup companies, that the field of rare disease therapeutics, especially for genetic medicines, hit a roadblock a few years ago. And it became clear that the criteria that are necessary for the biopharmaceutical industry to invest in and commercialize products, that the notion of a one-and-done treatment in a really small market didn't fit. We actually experienced a fair amount of support prior to that. And when pharma turns their back, investors turn their back. So we were sort of all dressed up and nowhere to go. Started 8 companies, had multiple products in the clinic, and everything ground to a halt. The rare disease communities call it the great abandonment in a very negative, almost sinister way. And I don't think it was sinister. It's just that the notion of genetic medicines, KJ, one-time treatments, AAV gene therapy, didn't fit the formula of the biopharmaceutical industry. So if I can share with you kind of a Venn diagram as the way, as where we are, and this sort of led me to making the decisions we had, that the biopharmaceutical industry really served us well for many diseases and been remarkably productive. But these constraints around market potential, sales revenue, growth potential, didn't really fit into many, virtually over 95% of rare diseases. There were a few Zolgensma and others that fit. But 10 years ago, that out of the window of the biopharmaceutical industry, it was rather small. But what's happened over the last few years, if you think about biopharma and what they'll invest in and what we can do in genetic medicine, the genetic medicine's potential is blown up. And it's not fitting the majority of what we can develop and finance through the pharmaceutical industry. This is enormous opportunity, but it's an enormous challenge. And we have to think about different models. And I think that's really what led us. I met with John before I met with our boss and said, I'm thinking about leaving Penn. And he said, why? And I told him why. He said, we're behind you. Because what we're about is not, as John said, patents and publications, it's changing the practice of medicine. And in my area, we had hit a wall. So at that point, we asked the question, is there a business model that may succeed and create a new approach that really leverages the advantages of rare diseases and one-and-done treatments? And we said, we think there is. And so the principles, the 5 principles that we founded our new initiatives on: outstanding, best-in-class science, but platforms. Platforms are the same approach to the treatment. You just change the gene or you change the guide. And if you can develop the notion of drugs as platforms rather than products, there's cost savings and you increase probability of success. Unmet need. Really initially focus on unmet need that paves the way for fewer headwinds. We have to be prepared to commercialize our products if pharma won't. And I think one-and-done treatments provide opportunities for that. And then ultimately partnerships. So what we've done is we've advanced private-public partnerships with governments in which the government is a payer to work together collaboratively, cost-sharing, risk investment, to develop products that we can sell at a more affordable rate, especially in middle and low-income countries. So where I see the world right now, and you'd probably not be surprised to say this, I think there's an enormous opportunity to develop genetic medicine for ultra-orphan diseases, without question. And let me describe why. So if you look at all of rare diseases, and I'll end with this, 10,000 of them, only a few percent would meet the criteria of a pharma investment, the larger market opportunities. But there's a growing movement who sees almost the moral injustice of not moving forward in diseases that fall below the line. And that has led to huge opportunities in the really rare diseases. A lot of the work that Peter had started at FDA and has continued under FDA and also in other health authorities, we have special considerations if the disease is very rare. Also, I'm looking at Ben, wherever Ben is. When we negotiate licenses for PCI, if we can argue that for their ultra orphans, the economics are different than if they're more common diseases. When we go to service providers like Franklin Biolabs who are here and our CDMOs, we can get a substantially reduced rate if it's an ultra orphan. So you put that together with diseases in which it's relatively easy with current technology to show a difference. There is an opportunity there, but the key is scale. We've got to scale it so the aggregate of the market opportunities are sufficient to justify the investment. And I am absolutely confident that's what we're going to be seeing in 26 as we and others start to see this opportunity. So when you look at all of rare diseases, we were only focusing on those that were the most common. But I do think for the least common, there's unique opportunities. But once you exceed that threshold, you sort of hit the wall again. And so it's really those diseases that aren't common enough to justify pharma investment, but are too common to justify the considerations that we're getting for the alternatives. And that's sort of the direction we're going.
Okay, we'll come back to that in a minute. And before I go on, I'll just ask one more question. So you mentioned that, when pharma abandons things, investors abandon, and therefore there has to be a different way. Sometimes the pendulum is swinging, though, and there is money poured into rare diseases. But now I think we're starting to see the pendulum move back away from rare disease. Do you agree with that? And at what point, is there anything that we should be doing, with, our Congress people to help think through how do we get more attention to rare disease?
Well, anything we can do. And I got to tell you, one of the incentives for any of us to invest and develop products in rare diseases, especially the most rare disease, least common, something called a priority review voucher, which is awarded to a drug developer with rare pediatric diseases that you can then monetize for $150 million. That is huge capital, especially if your cost structure is low. It's a game changer. That wasn't reauthorized. And it was incredibly successful. And it's still not reauthorized based on one person's vote. So go to Every Life Foundation, which is an advocacy group, reach out to them, and we're one vote away from regaining the priority review voucher.
Very
that would
important.
be enormously helpful because the potential of the voucher is attractive to investors.
Thank you. Okay, so we'll move on to Dr. Greenplate now, who's joining us by video. Allie, thanks very much for joining by video. So you've been at Penn since 2019, and I'm sure you're looking forward to your 30-year career there.
Absolutely.
I often say that, it's your generation that's going to make, tremendous change, of all the biology understanding that we've had in the last 30 years. Here's a great opportunity, for your generation to make tremendous changes in your career. And you've been focused on immune health, and you're bringing the technology of what I call, your generation to bear, computation, things like that I didn't know about, probably still can't do today, even though I would like to be able to. AI, you're bringing all of that to bear in your research. So what do you mean by immune health, and what do we all have to look forward to, and what are you looking forward to, let's say, over the next 5 to 10 years? Oh, and by the way, everybody can see you. You're projected on the big screen. and we can hear you great.
Perfect. Thank you so much. And thanks for accommodating me to be virtual. So yeah, this idea of immune health is something that I've been really excited about for about a decade now, and have been really lucky to be able to pursue this line of research at Penn. So the idea here is that if you think back maybe 20, 25 years ago to where we were with genetics and its role in precision medicine, we're at that same precipice now, but with the immune system. So the idea being that, 25 years ago, the Human Genome Project was finishing up, it was underway. We were sequencing the human genome, and along the way, developing new technologies, developing new ways to scale this type of information, and uncovering that data generated through these technologies actually told us something about the health of a person. And through that, there's been an explosion of precision medicine tools targeting gene mutations in cancer. Obviously, all of the work that the other panelists are doing targeting gene mutations in rare diseases. And where we are with the immune system now is that we are just about to capitalize on the ability of the immune system to tell us about the health of a person. And so what I mean there is that genetics tells us so much. It tells us the basis of what a person is predisposed to do and to be, and the impact of that on their health. But it doesn't always account for the lived experience of a person, the infections that you get, the environmental exposures that you have, the vaccines that you get. And your immune system is really the computer inside of you designed to take that information in and communicate it to the rest of the body. And when we say immune health, we mean, what is that history that's captured by your immune system? What is your potential for health, your potential for disease, your potential to respond to any sort of therapeutic based on that lived experience? So we're trying to decode the immune system to tell us something about your health, both you as a whole organism and your immune system as a system in your body. You can also think about it like if you need to quantify your heart health or your brain health. There are tests available for that in the clinic. The technology has not caught up quite so much with the immune system. We can really only enumerate main cell types. And so, why is now such an exciting time to be learning about immune health and figuring out how to implement it in the clinic? I think it really is tied very closely with the advent of AI and other machine learning tools. Unlike genetics, the immune system is more than a couple letters or base pairs. I don't mean to simplify genetics down to simply that. But it's at this time pretty complicated and widespread. In addition to the number of cells that you need to enumerate, there are features of those cells, the way those cells communicate with one another, the locations that they're found in the body. And that sort of information has not been computable up until now. There's just been too much information for the current computational structures to handle. With the advent of AI, we can now actually start leveraging all of these connections between the immune system and interpreting that into immune health. So if you think about the immune system, it's like a sweater. You pull a string and another one moves. We can now take that entire sweater as a picture instead of just individual strings. So what am I excited for in the next 5 to 10 years? I think that if you go back to the comparison with the Human Genome Project, I think that we are about to have a whole new wave of technology that is designed to scale our ability to read the immune system in a cheaper, faster, more accessible way. This has been done for decades in laboratories, and it's brought about huge changes in the immune system. But I think in order for AI to reach its power, you need much, much more data, and you need to bring the cost down much, much more to facilitate that. So I think we're about to see a whole new way of describing the immune system in a way that can be done outside of a R01 funded research lab. I also think that we're going to start seeing the use of the immune system to deliver precision medicine. So what does that blood draw that you get at your doctor's tell us about your ability to respond to any number of biologics? If you have rheumatoid arthritis, which is the first drug that you should go on? What one has the best chance of putting you into remission? Instead of doing the let's try out 4 or 5 in a row based on your physician's most educated guess. So I think we're really about to see this convergence of technology, biology, and clinical practice in the coming years, and I'm really excited to get to be even a small part of that.
Yeah, thank you for those comments, and I'm excited for you. In 2006, when I was diagnosed with ovarian cancer, and John mentioned the Clarity Foundation, I was so pissed off that I couldn't get my own tumor profiled. That's why I started this nonprofit to help other women get access to molecular profiling, to help decide what treatment would be the best. I had this rare clear cell carcinoma, and carbotaxol, I shouldn't even been on that, but I did go on it. I'm here to tell about it, so must have done something. But now it's in the NCCN guidelines that anybody with cancer, and of course, especially ovarian cancer, can get their tumor profiled. So when you talked about rheumatoid arthritis and what should you take first, why should it be just pick out of the hat? Why shouldn't it be tailored for you? I absolutely think that's coming, and great that we have people like you that are going to make that happen. So thank you. Yeah. Okay. So we're going to turn now to Dr. Marks. Peter, I don't think you need any introduction, actually. Many of us have worked with you when you were at the FDA, and for those of us who maybe didn't, we certainly came to know you during the COVID period, and so appreciative for everything that you did to really push pharmaceutical companies to speed up their work in the vaccines and to use the best clinical practice to bring to bear so we could have those vaccines sooner, and we really, really appreciate that. So Jim mentioned baby KJ earlier. Tell us about, from a regulatory perspective, your experience, and maybe go into a little bit of detail about baby KJ, what the phenotype was of the genotype that led to then this one and done approach, and why we should all be so optimistic about the future for these types of very rare diseases.
Yeah, so thanks, first of all, thanks for having me. I think baby KJ it's a great illustration of what's possible with technologies that have come together. It's also a great illustration of where we can go in the future with something we've already kind of alluded to by Jim of either platform or process-based approval. So let me just start. Baby KJ is a great example of a child born with a disease that was either had a 50% chance of killing the child or severely impairing the child. You knew that. You also knew if you didn't intervene relatively rapidly, you were going to lose a window to actually make a difference. And the beauty of having the confluence of whole genome sequencing as a possibility now, along with the ability to rapidly manufacture a potential therapeutic that could intervene through mRNA constructs, the ability to test that in an animal model. Now, we can talk now about whether you really needed that animal model testing, but they did in this case. And then to be able to produce it and administer it, and then be able to actually tweak that, administering it a second time to the child, so that at the end of the day, they were able to, in real time, tweak a therapy such that people may ask, well, what's happened to baby KJ? Does anyone know? You can see pictures on the internet the last pictures I saw, 16 months old, looking like a pretty healthy child walking around, no feeding tube. It's pretty amazing. So that's, I think, the confluence of what can happen when, when you put the weight of modern molecular medicine to bear, you can actually make a difference. And the question is, how can you make that difference scalable, right? And I think I would say that it actually means that, to me, the only way to make a difference is if we can make that into a commercially viable model. And the way you make it into a commercially viable model is to go back to the beginnings of biologics, when process was everything and it wasn't, process was the product. To me, what it means doing is allowing a little bit of risk that something might not work, or that something could go wrong that you did not anticipate, in return for the large majority of the time being able to actually treat people and end up with successes like baby KJ. What do I mean by that? Yes, idiosyncratic toxicities are always possible. Could you miss something that could be an off-target effect? Could you miss something there? Could it be something that's not targetable like you thought it was? That's possible. The problem is that we need to kind of turn our paradigms around. So one of the problems that you have as a regulator is that oftentimes we tend to have a lot of issues about alpha error, and we don't worry about beta error as much. In other words, if we don't want to make a mistake by a sin of commission, but we overlook our sins of omission. And unfortunately, if you're a parent with a child dying, you're really upset about sins of omission. And so we have to be able to balance that. And I think this concept of finding processes that we can do over and over again, and I think CRISPR and mRNA technologies, the manufacturing that goes into that, the basis that we're working on, because the nice thing about those 4-letter codes is that there's only so many things you can do with them, and they can be made reproducibly in terms of products. If we could find a way that you might not have one process, but you might have a series of processes that could cover rare diseases, some for mRNA, some for AAV, that you would follow the processes, and rather than going back to FDA every time for an IND and then a BLA afterwards, you basically have a BLA process that allows you to say in a qualified laboratory where you're comfortable. FDA inspects anyway. You basically inspect and make sure you're comfortable that everything from the concept of the product, how you get the genetic sequence you're going to use, to some type of testing, which might very well use AI because of what AI can do to test guides and targets, over on to how you manufacture this, could actually be in a BLA. And then you'd have a company that could do that for dozens upon dozens upon dozens of children at a reasonable cost, because they're not going back doing the regulatory work every time. And you'd probably want to also figure out how you're going to try to avoid having to do an expensive non-human primate study every time, whether that's by use of organoids or by skipping animal testing entirely. Would depend on the technology. But basically, the idea here is that if we're going to make this a reality for rare diseases, we're going to have to find a way to not have to kind of go through the toll booth in the desert. That's an allusion to a Mel Brooks movie, Blazing Saddles, where to really slow down the bad guys, they set up a toll booth in the desert, and they need to go back and get a bunch of nickels to get through it. We need to avoid having to put in a nickel every time we want to treat a new rare disease. Yeah. And we basically need to just open up the gate and let this flow, knowing full well that you occasionally look. Right now, and part of my perspective is colored as an oncologist. We give plenty of treatments where we know upfront we will kill 5 or 10% of the people. I'm not saying that that's what we want to do here at all. Please, no, that's going to get quoted, and Dr. Mark says it's good to. No, but we do need to be willing to say that if you have something that has 50% chance of lethality, we're going to actually figure out a way to make things happen, or even a 10% chance of lethality, that we're going to figure out a way to make this possible in a way that we're not right now, because there are just way too many diseases where not intervening is leading to tremendous, if not mortality. If you've seen children who are, a great example, because I became very familiar when I was at FDA with various children with lysosomal storage disorders, it is absolutely gut-wrenching to see. I mean, it's gut-wrenching to see what happens in multiple diseases, including Duchenne's, et cetera. But if you have a child who's developing normally, and then you see them fall off to the point that they can't communicate with you anymore in the course of a matter of a year or two, it's gut-wrenching. So those parents are looking for a better path forward. To date, it's been very hard to get there, but it should be possible if we did this in a process-based method, because the current method of going through and making each new product a new birth, again, it is like the toll booth in the desert, because you have to go through a lot of different things. Now, just yesterday, FDA released some nice guidance on the web. They made an announcement that they're going to make this easier, that they won't require the same number of qualification lots, manufacturing lots or they may not even require manufacturing lots at all for rare disease products. In order to help move this forward. Let's see, hopefully they'll put the money where the mouth is, and we'll see this move along, but this concept of process-based approvals could really be transformational.
I'm going to have a follow-up question on this, but just to say that we're going to go to audience questions pretty soon, so be thinking of your questions, and you can just use a microphone on the table there. So follow-up, we're in a new world now. You're not there. You're at Eli Lilly. You're going to try to make things happen in the new FDA. Do you have any advice for companies or the people that interact with the FDA about how do we approach in this new environment? What do you see as some of the challenges now?
So everything I ever learned that I needed to know, I learned early on when I had my first pharma job, which is you always want to have a primary strategy, but in the closet, you also want to have a backup compound. And so I think the primary strategy here is you go to FDA and you try to make this happen. You try to hold them at their word. You go to them with a strategy, and you try to pursue it in that manner. Holding them accountable for doing what they say they're going to do. Because if they put it out in guidance, which is what it is when they put it out on their website, technically it should be guidance documents, but it seems like right now posting on the web is their moral equivalent of guidance. If they say that, we hold them accountable to it, and your attorneys, your food and drug lawyers, should hold them accountable to that. Now, that's the primary strategy to move forward. If that doesn't work for you, then you have to have a backup strategy. And the backup strategy is you're going overseas. Mm-hmm. And I think if enough pharmas start going overseas, China's lovely sometimes. MHRA is open for business. They'd like you to know that, by the way. I'm not being paid by MHRA.
And HRAC in Australia is too.
And TGA would love you to hear from you as well. You just go overseas and you get the job done there. Now, that's hard for rare diseases, right? Because these kids are here, and you can't send them there.
But you don't see a world where the FDA could evolve to have like a TGA, HREC style or an IIT style?
I'm hoping that I'm hoping that this idea of process-based approvals is something that they can take on and make into a reality. And I'm hoping that they can see over some of the current shortcomings that may be present to get there and rise to the occasion and say, hey, we can see we can put in place what's necessary to get there to process-based approvals. It's not simple. Just so you know, this is kind of Regulation 601, not 401 or even or 201. It's really advanced regulation of figuring out how you put the right controls in place so that you can have something happen on its own by a company repeatedly, time after time, with the right reporting after the fact, so you feel confident that like catastrophes aren't happening on your watch, yet you feel like you're actually allowing the ecosystem to bring benefit to more and more children.
Thank you. Questions from the audience? Yes, right here.
Out here. Yeah, this is kind of a question. It really touches on all the panels. Outside of genetics in orphan diseases, there are a number of ways that you can develop, create a development program that leverages orphan diseases. And so in immunology, for instance, T cell markers, so T helper versus T inflammatory cells. And if you take eosinophilic esophagitis, look at all that sort of stuff, and look at the clinical outcomes, and look at eosinophils, you can translate that to rheumatoid arthritis. So to your point, Dr. Wilson, then when you go to somebody who wants to potentially invest in a partner, and they say, we've proven the concept of EOE, but it'll work in rheumatoid arthritis. And I think, Dr. Marks, what you're saying is that the FDA's become much more receptive to biomarkers, less animal studies, and so you have these orphan diseases within your broader development program, and it allows you to sort of extrapolate into those bigger populations for investment.
Right. If I could take a crack at that. Yeah, the question was, is there a drug development strategy where one begins in a rare disease, maybe a simpler pathology, easier to show proof of concept, and then leverage that for more common diseases? And so that's an argument we pitch all the time to try to get investment in rare diseases. And there are examples of that. For genetic medicines, I believe that promise is what led to companies such as Takeda and Pfizer and others to lean in about 5, 6, 7 years ago into rare diseases. The problem with genetic medicines is well, scientifically that translation may be relatively obvious. There's a different risk profile in more common diseases than there is in the ultra-orphans, where there's almost any risk may be tolerated. And there's an example that is held up in front of me as to why I shouldn't invest in rare diseases, the commercial viability of gene therapy, and it's the hemophilia A and the hemophilia B approved products. Incredible. Inject once, you have your factor 8 and factor 9, don't have to get the infusions. Remarkable. But in the US, where the standard of care and access to long-acting factors is available, that it's approved, it's priced, but no, let's let docs prescribe it and the patients accept it, it's not going to be commercially successful. Remember I said my third pillar was unmet need? I think we have to kind of stay and make sure we focus on unmet need as we try to spread our wings in the way that Peter had said. Now, what happens in a pharma company when they try to identify extrapolation opportunities in which are more common, there may be other treatments, and the risk-benefit calculus, while it may have passed the gate of regulators and even payers, it was fascinating. But just because factor 8 and factor 9 didn't succeed necessarily in high-income countries, that doesn't mean it wouldn't be enormously helpful in middle and low-income countries where they don't have access to that standard of care. So. Maybe,
Ali, you would want to comment. So it was really, the question also included the disease of eosinophilic esophagitis translating to a treatment for rheumatoid arthritis. And I can imagine that, you might think that there'll be a biomarker one day to tell us how to think about diseases being very similar, even though they look very different. Just like, if you and I both had ovarian cancer, they might be treated differently, but if I had ovarian cancer and Jim had lung cancer, they might be treated the same because of the molecular biology. So maybe you would want to comment on the idea of biomarkers and the approach.
Yeah, absolutely. I think that's a great point. In immune health, we often think of the immune profile as disease agnostic. So one of the things that we're doing is building a large language model based on immune profiles from thousands of different patients who have come for care at Penn. And the idea here is that there are biologics and other drugs that are used across different diagnoses. And so instead of giving a treatment based on a diagnosis, might we actually think about your immune state being more important than the diagnosis itself? I think one of the key things to think about when it comes to biomarkers is less about single cells or the presence or absence of single cells, but rather what are the relationships in the immune system. So it's a little bit more complicated than measuring one cell, one protein, but do those relationships in eosinophilic esophagitis and RA tell you that the person had the same history and the same propensity to respond? And if they do, then it's perhaps likely that the same drug could have benefit for both of those subjects. And so ultimately what we're trying to do is to take a look at human health and disease outside of the context of disease names, and instead of thinking about your immune system as the general barometer for health and disease.
Thank you. I imagine your databases are incredible, and that you're going to be mining them gaining lots of insight. So looking forward to that. Just do a quick time check. What do we have, 5 minutes maybe? Oh, okay, let's take another question.
Hi, my name is James. I really appreciate your discussion on rare diseases. I'd like to bring the conversation to mental health diagnoses and health policy. So I'm currently building and fundraising, by the way. I'm currently fundraising for my behavioral health software startup that's aligned with Medicare reimbursement and private employer benefit after personally being diagnosed with bipolar 2 and borderline personality disorder late last year. So my question is, what CMS coverage or reimbursement change would be necessary for precision medicine to scale under Medicare and Medicaid, especially in this current administration and possible continuation of the next administration?
Yeah. So I'll try to take this. So I think what you're talking about is what has to happen here for this process-based medicine approach to come, is that there will have to be, CMS will have to come along and consider things that are process-based approvals or things like this to be accepted for reimbursement. I think there again, traditionally we've had this process where you've had to have effectiveness information for approvals through CMS. Here, I think what you'd have to say is that the process is, we're sufficiently confident in the process that CMS is willing to reimburse for it in order to have this become commercially viable. I guess that applies to this type of an idea too, that you would have something that CMS would, it would be a process or a program that they would hopefully be able to reimburse for. This just goes to something I wanted to say, because I think Jim got this exactly right, which is that we need to understand that if we really want this to work, and I think to grow upon, the right way to think about it is crawl before you walk and walk before you run. Let's go to the really rare diseases that affect young kids, where the risk-benefit is generally really clear. Show that this can work there. Then we can squeak it out to larger populations and larger populations, showing that this process base works. It also is good for the federal government, because if they know that they're dealing with smaller populations and not really giant populations, it makes things a lot easier. The reason why there was so much panic over the Alzheimer's drugs is because there are a lot of people with Alzheimer's out there, and the dollar figure becomes astronomical really quickly. Whereas, 100 or 1,000 baby KJs, that's not going to break the bank. And if we can show that that really works well, and there are 5 or 10 processes that can treat 1,000 baby KJs, that actually would be something that maybe we could get CMS to say, okay, well, each one of these costs $200,000, $300,000. Cheap at the price, because otherwise institutionalized child for 5 years costs a fortune. So that's where I think you try to work through this, and it could make a big difference.
Thanks for those comments. And also just to acknowledge that we're still, we haven't even thought through how to personalize behavioral health. And we do have to do that too, because there's so many different diagnoses where we're just not helping people, and there is huge unmet medical need. And lots of, if you do like an all-comer trial in any type of behavioral indication, you're going to fail, because on a population basis, it just doesn't work. So we do have to figure out how to individualize that. So thank you for bringing that up, and good luck in financing your company. I think we are a little bit over time. So Lori, I'll turn it over to you. And thank you very much to everyone. Thank you.
Yes. Thank you again to our amazing panel, Dr. Epstein, for being here. The amount of expertise and leadership we were able to convene today is really amazing. And I hope all of you got a sense of the strong team and commitment we have at PCI and Penn to commercialization and partnership. And despite maybe some changing times and turbulence ahead, we're ready and active to be creative and meet the moment. And part of why we come here is to encourage all of you to follow up with us, come talk to us, come visit Philadelphia. We were just named the #1 place to visit in 2026 by both the New York Times and the Wall Street Journal, because we're celebrating the 250th anniversary of our country. So not only can you find out about all this amazing innovation, but it's a great place to be. And before I hand it over to our wonderful sponsors who have partnered with us every year that we've done this, and also an extra thank you to Dr. Wilson, who I think came out the first time 10 years ago when we had about 30 people standing outside with you and Carl June. I wanted to highlight, we have outside a list of over 40 startups from Penn who are raising capital right now. We will be posting this on our website, but please take a look. Talk to any of us at Penn. We can put you in touch with the founders, and these are all real-world examples of faculty innovation underway that we are supporting. So please take a look. And without further ado, I just wanted to bring up Catherine Doyle, a partner at Saul Ewing, and an amazing IP attorney who's been part of our work at Penn for a long time.
So I'm the last thing standing between you all and the fun out there, so I'll make it really fast. It's always a pleasure for Saul Ewing to be asked to participate in this event. I think of it as one of the most innovative events that happens at JP Morgan, personally. I don't know how you all feel, but that's how I feel. My colleagues, Domingo Silva, Justin Crotty, and I, delighted to be here. I want to thank PCI and Penn, because it's amazing what Penn has done over the last many, many years that I've been working with them, but particularly over the last 10, 15 years in terms of being innovative in how the research that Penn manages to do has translated into real meaningful therapies in the clinic. And then lastly, I want to thank the panel, because that was one of an amazing, enlightening conversation, and one I learned a lot from. And so thank you very much. So please join me again in thanking the panel.